Hello, everyone. My name is Marvin G., and I'm head of target discovery at 3T Biosciences. We're a company focusing on identifying novel targets and developing therapeutics against them for the treatment of solid tumors. Today, I'll tell you why we're interested in developing the next novel targets for T-cell therapies, what our process is, and our progress thus far in this space. Where I'd like to start is by talking about how T cells are integral to anti-tumor immunity. The life cycle of anti-tumor immunity begins with an initiation event, whereby the immune system can present cancer antigens expressed by the tumor, and T cells can then recognize these antigens presented by human leukocyte antigen, often referred to as HLA, and these T cells are then primed and activated in the lymph nodes. T cells can home and traffic to the site of the tumor to then kill tumor cells upon recognition of this peptide HLA complex expressed by the tumor. This important recognition event through the T cell receptor, or TCR, allows this cycle of tumor elimination. What we're focusing on 3T is this particular event and looking to identify TCRs and their targets. The interest in targets has been stimulated by the incredible responses seen in recent years by the use of checkpoint inhibitors. We've seen durable responses with 34% overall survival rates as far out as 5 years in melanoma, shown on the graph on the left, and now furthermore in many other solid tumor indications with remarkable results shown on the right. What we're finding in these treatments is that checkpoint helps take the brakes off of the T-cell immune response, certainly among other changes. Now, the question is, what are these T-cells recognizing? And as a means to discover new targets and avenues of therapies, what we're focusing on are patients that are responding to their tumor. One can hypothesize that the right immune response is not generated in some patients to target their cancer, while others do have that specificity. By focusing on patients that are responding, we're able to identify TCRs and their targets that enable for tumor elimination. And our goal at 3T is to use this information to help provide productive responses to as many patients as possible. Now, this approach has been well validated by recent and ongoing trials utilizing T-cell receptors and adoptive T-cell therapy that recognize cancer antigens, such as NY-ESO-1 and more recently MAGE-A4. Now, recent ASCO data has shown that T-cell therapies targeting MAGE-A4 have produced significant overall response rates in a variety of solid tumor indications of high unmet need for hundreds of thousands of patients. While these are early results, it's very promising for the field of T-cell therapy and targeted therapies in particular. Now, of course, certainly this field has existed for some time, and there's a number of targets that have been identified over the years. And studies of T-cells resident in the tumor have revealed a diversity of targets, including bystander viral antigens and, of course, tumor-related targets. Targets like NY-ESO-1 and MAGE-A4 fall within a class of tumor-specific cancer testis antigens, named so because they're often only expressed in development and restricted to the testis tissues. Other antigens are being pursued, such as neoantigens, which are mutated sequences that are often, however, patient-specific and thus hard to target on a broad basis. There are well-known tumor-associated antigens that have preferential expression in tumor over normal, and differentiation antigens that are restricted to particular tissue type. While these different classes exist, the universe of potential targets is actually quite vast compared to the dozen or so targets that are known to date. And our goal at 3T is really to identify as many novel targets as possible to really broaden the patient population that we can help treat in the solid tumor space. When we ask the question of how we could develop technology to find the best novel targets, we designed the 3T Trace platform. which stands for T-cell receptor antigen cross-reactivity engine. We focused on developing a platform that we could use to identify the specificities of T-cell responses from patients responding to therapy in an unbiased way, to discover those that would be the most immunogenic and broadly expressed across patient population. In developing this platform, we knew that the TCR itself, the therapeutic modality, would be important to develop as well, and that we can use this same platform to guide the development of therapeutics targeting these novel targets from a safety and efficacy point of view. What makes us able to use our novel technology are several advances in the field, including the identification of elite responders from checkpoint inhibitor therapy. These productive immune responses serve as a source of high-quality TCRs and targets that could be applied to a broader patient population. In addition, single-cell sequencing technology has allowed high-throughput identification of these TCRs from the T cells infiltrating the tumor, as well as from the periphery. And lastly, the 3T trace platform is enabled by a high-diversity, high-throughput yeast display library that displays billions of different targets, coupled with active machine learning approaches that can accurately identify the specificities and potential off-target cross-reactivities of TCRs and TCR-like therapies. 3T Biosciences is based off of over 20 plus years of research and discovery research from the lab of Christopher Garcia from Stanford University. With this, we've built an industrialized platform to identify targets and therapeutics. Importantly, we focus on an immune response guided target identification methodology by starting with patients with productive immune responses. We believe we have the most comprehensive TCR cross-reactivity screen coupled with the best-in-class TCR target discovery engine. This helps us ultimately identify the best TCR compounds from checkpoint inhibitor-treated patients in order to develop therapies. I'm joined at 3T Biosciences by an experienced management team, including Hans-Peter Gerber, who has had previous experience at Pfizer Genentech and Seattle Genetics and previously led drugs to IND approval and through the clinic. As well as Glenn Dorado, who's had previous experience completing deals totaling over $2 billion. And of course, we also have our fellow co-founder and co-inventor of the 3T Trace platform, Leah Seiberner, to lead research efforts. We've come together as a team to take early research and discovery processes using the 3T Trace platform into the clinic to develop the best TCR-based therapies. We're also joined by a proven scientific advisory board, having leaders like Christopher Garcia, Stephen Schoenberger, Mark Davis, and Javier Chaparro-Riggers, who are leaders in their respective fields of TCR peptide HLA, structural biology, systems immunology, vaccines, and antibody-based therapeutics. We're happy to have these leaders recognize the value of 3T Trace platform in developing the next generation of therapies against novel targets. In the next section, I'd like to dive into the specifics of the 3T Trace platform and how we use it to identify novel targets. Importantly, as I mentioned before, one of the important facets of the technology is focusing with the end in mind. And what that means is starting from patients where we know that there are productive immune responses and profiling their immune responses on the 3T Trace platform to identify the most active TCRs and the most prevalent and immunogenic targets. And as I said before, we're able to use this platform to continue to guide and engineer the specificity of these molecules for safety purposes. And that's in comparison to conventional approaches, which have largely started with a particular target in mind, trying to develop a reactive TCR, one that's potent and safe. And with our methods, we believe by starting with the patients, we can really go after and identify quickly the most robust immune responses. So to break it down into a 4-step process, what we do is we focus on starting with the patients by sequencing and profiling their immune responses, primarily their T cell responses, as well as the tumor biology. From there, we identify TCRs of interest using sequencing information and screen it on our proprietary 3T trace platform to identify the landscape of potential targets that can be recognized by a TCR. Coupled with our active machine learning algorithm, we can identify synthetic peptides that can be recognized by the TCR and then accurately predict the human specificities and potential off-target cross-reactivities that may be liabilities in the clinic. Lastly, we validate our targets through antigen processing of our targets, as well as T-cell activation and tumor cell killing. To speak a little bit more about our platform, it's a yeast display platform that can able to display billions of different peptides presented by a particular HLA. On the right in orange, we've listed a number of HLAs that we've been able to develop on class 1 libraries, as well as a number of class 2 libraries. What's important is that we've also been able to profile a large number of patient samples spanning across a number of different solid tumor indications. As I mentioned before, we focus on looking at patients that are responding to therapy and have developed an internal database of over 140,000 TCRs coupled with public data to identify TCRs of interest to discover their targets. And to speak a little bit more about our computational approaches, we've designed machine learning algorithms and computational pipelines that can accelerate analyses from 2 weeks down to 4 hours to really identify as many targets as possible from our pipeline. We also use computation to design our libraries, prioritize T-cell receptors we're identifying from responder patients, characterize the targets that we're identifying, and profile TCRs for specificity. Now, the platform itself we've validated through many different facets, one of which is surveying the landscape of TCRs that are targeting clinical antigens now currently being used, as well as the variants of these TCRs. On the right, you can see a number of different targets where we've been able to actually identify and confirm our ability to screen TCRs that recognize these antigens and confirm their specificity by our approach and algorithms, as well as actually identify potential off-target toxicities that may be liabilities in the clinic. What's interesting about our approach is that we can identify cross-reactivities that are actually not sequence related and thus make it very difficult to detect by conventional approaches. And for example, can identify potential normal tissue toxicities based on these cross-reactivities. And here's an example of what we often see when we screen TCRs against a known target. Here I'm showing you 6 different TCR variants that are known to recognize the same target, highlighted in cyan, compared to other potential unintended targets from the human proteome. On the y-axis, we're representing the probability of these TCRs to bind that target, and on the x, the different targets possible. And so what's interesting is that we can compare TCRs relative to one another to identify the good, the bad, the ugly. So TCRs that have high specificity like TCR1 and 4 versus those that have poor specificity like TCR3 and 6, despite all of these TCRs known to be recognizing the same target. And this is important to really prioritize potential candidates going into the clinic to ensure the best safety possible. What's also important about the 3T trace platform is that there are multiple quality shots on goal. I mentioned that we have priorities in developing TCR-based T-cell therapies, but there's also opportunities to develop peptide vaccines from the synthetic peptides we can identify from the 3T trace platform itself, as well as monitor other kinds of protein biologics, including TCR mimics. For example, IgG-based scaffolds that can recognize peptide HLA. It's quite important to be able to monitor the specificity in order to choose candidates going into the clinic. And also importantly, this is an unbiased platform. Any disease that involves T cell responses, we can measure their specificities as well as potential off-target toxicities. Our primary focus is in oncology, but we've also been able to explore avenues in autoimmunity, allergy, and viral infection. So next, I want to move into what we've been able to identify with 3T Trace platform. So starting with a response-guided target ID approach, we had to develop a number of criteria to represent what is a productive response. And really the goal here is to identify targets that are abundant and immunogenic. So here on the left is a list of parameters that we use as filters to help us prioritize TCRs of interest, including TCRs that might be shared across patients, as well as those TCRs that are enriched in checkpoint inhibitor responding patients. We look at classical markers of immunological activity like clonal enrichment of these T cells in the tumor microenvironment, as well as markers of T cell activation by gene expression signatures. What I'm showing you here is our list of targets by program ID, as well as a number of parameters that we look at to prioritize our targets, some of which I mentioned before, including clonal expansion of the T cell receptor at the site of the tumor, the clinical outcome of the patient highlighted in blue, whether or not we're finding these TCRs across patients designated by public TCR, and then a number of characterizations around the target, including whether or not it's a cancer testis antigen, meaning it's highly specific to the tumor, and the tumor over normal expression by RNA to identify potential disease indications that might be preferentially useful for these targets. Lastly, what's important is the epitopes we've discovered have not been previously described, but the proteins to which they map to have been described to some capacity, even some of which are known as driver genes in terms of driving tumor growth, which is important from the aspect of tumor heterogeneity and loss of antigen. And so that the more important that these antigens are to the tumor, the less likely that they will be edited out. Here I'm highlighting our top 9 cancer testis antigens in comparison to some of the leader targets in the field, including NY-ESO-1 and MAGE-A4. What's interesting is that we see a diversity of expression by RNA in a number of different tumor indications. And what's interesting is that we do see in our targets high and broad expression across tumor indications, which may signal that these are going to be valuable targets in terms of their utility in these different indications and be able to target a broad patient population. This is our pipeline to date, where we've developed a number of programs, including lead optimization of TCR-T, TCR mimic program against a known cancer testis antigen, as well as moving forward novel cancer testis antigens and non-cancer testis antigens into our pipeline through multiple therapeutic modalities. So I also want to highlight our ability to use the 3T Trace platform to actually guide and identify TCR therapeutics and or mimics against the targets of interest. So we can use this platform to identify TCRs that are going to be the most specific. On the middle panel, I'm showing you we've identified TCRs from patients that meet the benchmark criteria in terms of TCRs known in the field targeting a known cancer testis antigen, finding TCRs that are similar in potency against the specific target. On the right, we're using our 3T Trace platform to actually measure the specificity of these different compounds in comparison to the 2 benchmark T cell receptors. What we can actually see is that our TCRs we're profiling, we can actually see that they're more specific for the target, the intended target, with less liability and potential off targets, shown by the lack of color in the heat map. And when we use this information to look across the ability of a TCR to recognize different positions of a peptide, we can rationalize that our TCRs are more specific because they actually recognize additional positions of the peptide rather than a single hotspot. And this makes the TCR more specific for the target than, for example, the benchmark. We've looked across a number of different TCRs, both known, both TCRs that we've identified at 3T, in addition to other therapeutic modalities known as TCR mimics. These can be IgG-based or non-IgG-based scaffolds that recognize these targets. And we actually can see, using our platform, a variety or diversity of specificities for these different compounds. And this is quite important in terms of developing therapeutics for developing the safest compound. And so we see that the closer you are to the top of this graph, the more specific you are for the intended target. And it's not dependent on whether or not it's a TCR mimic, but rather how you develop these compounds and how you derive them. In summary, I'd like to leave you with our information in terms of our focus with the 3T Trace platform, that we've developed a best-in-class comprehensive TCR screen, as well as a best-in-class target discovery engine to really push forward the next wave of therapies targeting solid tumors. Importantly, we're focusing on a response-guided target identification method by focusing on elite responders in order to make this possible to treat a broad patient population and make as many patients responding as possible. We're looking to expand our capabilities not only in oncology, but in other indications as well. With that, I'd like to thank the 3T team and leave you with my contact information. Thank you so much for listening to the presentation.